welcome to the Blakemore Lecture Theatre in the Sherrington Building in Oxford. And this is the Oxford Autumn School in Neuroscience. And my name is Masood Hussain. And it's a pleasure to actually try to do this hybrid event, but I'd also like to thank Nick Irving for all his sterling work in putting this together. So we've got a very diverse and unusual package of symposia over the next 2 days, and I hope you enjoy most of it. We're going to start off today with something that we haven't done before, which is a symposium looking backwards and looking forwards to where we are now, because it turns out that this is an important anniversary for several Oxford luminaries. And to begin with, we're going to discuss Richard Burton, who wrote The Anatomy of Melancholy. We're going to look back at depression 400 years from now and what we know about depression now. And secondly, we're going to go to Thomas Willis, who was born 400 years ago today and perhaps was the first clinical neuroscientist in the world. And we're going to look back at his work and also look to see where we've come in those 400 years, at least with aspects of vascular neuroscience. So to kick off our first symposium, which is going to be looking back, looking forwards, I've got Professor John Geddes. John is Professor of Epidemiological Psychiatry here at Oxford. He's also fellow at Merton College. And he was telling me that he has been head of department, but this is his final day. So I don't know whether you're looking forward to that, John, or looking back in sadness for having to leave it. But John has explored the work of Richard Burton, who 400 years ago published The Anatomy of Melancholy. So perhaps, John, I can get you up here and then you can tell us a little bit about that. Just find your talk.
Thanks very much indeed, Masood, and what an amazing pleasure to be here talking to real people. Actually, today I've got about 3 of these, so we've kind of gone from 0 to nothing, and let's hope that continues. And I'm going to be talking about Robert Burton and The Anatomy of Melancholy, really to look at a bit. The thing about The Anatomy of Melancholy, if anyone has looked at it, is that you'll know that its final edition, around 1650, it's 500,000 words, and it's an enormous text. So I'm definitely not going to summarize it in the few minutes that we've got, but let's see where we get. And now what I want to do is explore its current relevance, really, and give you a bit of an idea about it. If I can just get the... there we go. So this is the very famous frontispiece of The Anatomy of Melancholy. So the first edition is 1621, and there is Robert. That's the portrait that is usually in Brasenose College, and at the moment is in the Bodleian. And I'll come on to why it's in the Bodleian at the moment. But I would recommend that if you get a chance, go and look at that portrait, because you get a sense of the fun in this man's eyes. When you see it face to face, you really get sucked in and just see the mischievous sense of fun and joie de vivre in Robert's eyes. This is the first frontispiece of the last Jan. The Anatomy of Melancholy, Burton first published in 1621, and he continued to revise it for the rest of his life. So that's for about another 30 years. And at the end, it's 500,000 words. And it is an intensely structured book. I won't go through the structure. This is the synopsis. And it would take quite a long time, more than I've got even to work through the structure. But essentially, Burton is looking at the causes and treatments of melancholy, referring back over about 2,000 years of unsystematically reviewed evidence, hence part of my title today. But Burton, although I think Willis located and his successors located the brain as being the place where the thinking happens, Burton did say, fortunately, the brain is the most noble organ under heaven, the dwelling house and seat of the soul, the habitation of wisdom, memory, judgment, and reason. So he get part right. He was writing in a time when melancholy or feeling low was actually quite fashionable. This is Durer's De Melancholia from 1541, incredibly influential. This is, I think, the most written about engraving in art history, portraying a figure of melancholy. And Burton specifically bases a lot of his thinking around this. Oxford at the time, this is a picture from 1605, was a very different place. You won't be able to read that, but you'll see a lot of the colleges are already there, but there's an awful lot of green space around. And Burton was a well-known figure around Oxford. He was involved in founding, we think, the Oxford Botanic Gardens, which at that time was the Physic Garden, which was set up because the Regius Professor of Medicine wanted a place to get the herbs and plants for teaching medicine from. So we think he was involved in that. We found some evidence that that was the case. And he was a well-known figure. So he was known as being very cheerful. Now, I'm just going to kind of allude a little bit to some of the environment at the time in Oxford around 1621. And one of the things that was happening in England at that time was that we were beginning to really drain the wetlands. And this has led over time to reduction in the peat in the fens, so that the carbon dioxide's gone into the atmosphere, leading to global warming. At the same time, there was 1625, so a bit later, but there was endemic plague. And it's quite interesting, kind of from our current perspective, just to look back at some of these big issues. There was a lot of concern about draining the wetlands and the effect that it would have on people's lives and climate and things like that. At the same time, there was a lot of concern about endemic plague, which had been raging for years and continued to do actually until the sort of Great Fire of London, when it sort of went away to some extent. But the 1625 epidemic, and plague was around all the time, but kind of, you know, just like our current pandemic got worse at various times. 1625, there were 40,000 deaths in London alone, massive exit from London, magistrates, doctors, clergy, all the people you need, legged it. And in fact, the king at the time moved from London to Christchurch in 1625. So that was Burton's College, where he was a student, and Parliament, in fact, was adjourned to Oxford because of the plague at Westminster. And although Charles and the court were welcome to some extent, there was no official reception because people didn't want to catch the plague. So all sorts of social distancing and restrictions at the time. So, you know, there are some similarities between what's going on then and what's happening now. You know, and it's interesting just to sort of muse about the Wellcome's current priorities, which are mental health. So that's Burton, infectious disease, plague, stuff like that, and climate. And there's this huge change in the English landscape occurring because the east of England, which was largely wetland, is becoming drained. So just another sort of interesting little reflection on how things have not changed or have changed, or the resonances between 400 years ago and today. Burton, I mean, I struggle to find much mention of him in the psychiatric literature because he's not a psychiatrist, he's a clergyman. He comes from outside medicine, but he's had enormous influence on literature. If you look at the lists of the great books over the centuries, they always nearly always include Anatomy of Melancholy, and had a major impact on Samuel Johnson. Lawrence Sterne actually plagiarized him. John Keats was specifically influenced by some of his great poems like Lamia, Borges, Samuel Beckett, Anthony Powell, and of course, most recently, authors like Philip Pullman have expressed their huge enthusiasm for Burton. So in a way, he's a dominating figure in literature, which is really interesting. And you will, if you do engage with the book, you'll understand why that it is, because the creators of encyclopedic fiction love the structure of Anatomy of Melancholy. The influence on medicine and psychiatry is less easy. And again, he's not writing from within our tradition or our discipline. But it is there, and indeed, Sir William Osler, who's one of our great, we have a lot of great Regius Professors of Medicine, but Sir William was one of them at the turn of the century, came over having founded Johns Hopkins University with colleagues and was recruited to Oxford. Osler was also the chap who invented modern approaches to bedside teaching of medicine. And he spent his last years in Oxford, and he became very, very fascinated by Burton. Wrote a lot about Burton. What did he see in Burton? Well, he saw a lot, but I'll just put a couple of quotes up. So he thought that the Anatomy of Melancholy is a great medical treatise, orderly in arrangement, serious in purpose, and weighty beyond belief. So it's not a comic book or a rambling book. It is a weighty, serious text. And that's Osler, who's not a psychiatrist. He's a sort of general physician. And in it, a subject of universal interest is enriched with deep human sympathy, so melancholy. And with its roots in this soil, Burton's book still lives. So what is it that Osler and many of us see in the Anatomy of Melancholy? Well, there has been an increasing, over recent times, increase in the number of writers who've written about their own experience of depression. One of the first of the modern crop was this fabulous book by William Styron, published probably about 30 years ago now, Darkness Visible. It's a short book. It describes Styron's own experience of serious depression. And at the time, I remember this being published, and it had a huge impact because it began to break down some of the barriers of talking about mental health, depression. I was very fortunate to work with Lewis Wolpert on this, but Lewis Wolpert was a famous cell biologist. Jim Davis, who was the head of science at the Wellcome Trust, was Lewis's PhD student, and Lewis died just earlier this year. But he too suffered from a depressive episode and wrote, and then actually recurrent depression all the way up to the end of his life. And he wrote this book, Malignant Sadness, but look at the subtitle, it's The Anatomy of Depression. And he was specifically influenced by Burton in this. And he wrote this book to find out as much as he could about depression, to see if that helped him. And that is precisely why Burton wrote The Anatomy of Melancholy, because he calls himself, I am Burton, I speak of melancholy because I am a melancholic. And Burton wrote about melancholy because he wanted to become expert, but also it became his active purpose. And by writing about this and having this focus, that protected him against depression. He took a very broad approach, and I'm not going to go into the contents of the Anatomy of Melancholy, but just to say that the most remarkable thing to my mind about the Anatomy of Melancholy is the breadth of the approach. So he includes looking at reading, walking, gardening, all sorts of activities that can be protective against depression. Of course, we're beginning to rediscover some of these approaches to depression. There's a lot of interest in social prescribing. And Daisy Fancourt and her colleagues have done some great work saying, well, what is it about these activities? What brain mechanisms do they kind of tie into? What are the underlying mechanisms? Because actually, if we could work those out, then there may be a whole load of other approaches to helping people with melancholy. That's going to be incredibly important because depression now, as we know, is probably the most important cause of disability globally. And we need as many solutions and as many easy solutions that people can manage themselves. So the idea of the expert patient is what, to my mind, Burton is. And that's an incredibly modern concept. So I'll finish there. I just want to highlight one or two additional points. And that, as Masood said, it is the 400th anniversary this year of the publication of the Anatomy of Melancholy. And we have just launched an exhibition in the Bodleian Library, which opened just yesterday? Yeah, yesterday. So it's, I was one of the curators, so I definitely recommend it. Full of very interesting materials from the vaults of the Bodleian, and also highlights some of the modern relevance of the anatomy of melancholy to current thinking and approaches to depression. We launched it. We had a very interesting panel, which is now live on the YouTube Bodleian site. So you just Google Bodleian and anatomy of melancholy, it'll come up. Really lovely panel. Katie Murphy, who's an associate professor in English and whose life's work has been the academic study of the anatomy of melancholy, who was my co-lead curator on the project. Sir Philip Pullman, who is the author of His Dark Materials and is a resident of Oxford, and very interesting, I think, listening to him about what it is about the anatomy of melancholy. Sister Stephanie Therese, who's lives in a convent in Oxford, suffers from bipolar, and again reads Burton from the point of view of the person who's experienced mood disorder. And a very interesting panel chaired by Niall Boyce, who's the current editor of the Lancet Psychiatry, who is currently doing a PhD in early modern works. So of course he is. So he is both a sort of editor of one of the leading psychiatric journals and interested in that area. And finally, we did put together, if people want to know more and more about the kind of themes of anatomy of melancholy, and explore some of the current research and interests in those areas. We put a series together. Ruth Abrahams, who was the communication officer in the Department of Psychiatry at the time, put together a really nice series of programs that were on Radio 4. I think there's about 12 of them, 12 episodes, only 15 minutes each, exploring different aspects, and that's still available on the iPlayer. I'll finish at that point. Thank you very much. What a delight again it is to be talking to real people, and of course into the virtual, wherever you all are. Thank you for listening.
Thanks so much, John. Maybe we could just hold you back for 2 minutes if there might be a couple of questions, anything anybody would like to ask. So what about the character, Burton? Tell us a bit more about him. You said he looks so happy in that portrait. Was he always happy?
Well, he says that he wasn't, and he did experience periods of melancholy. Yeah. And I think this is one of the reasons that Sister Stephanie Therese, and probably I, think that he may have had variable mood. Right. Contemporary accounts of his character are that he was very cheerful. He had, now at the time, being called facetious was not current way that we would see, which is a sort of slightly negative comment. It meant that you had a kind of, you were cheery, you kind of made light of things. And that was the way people reported his personality a lot of the time. So I think he was very engaged in Oxford. He was busy running around setting things up, but he obviously had periods of low mood as well. So I think, you know, the thing that comes out of reading The Anatomy of Melancholy is actually that it's a hugely fun text. It's very digressive.
It's 1 million words, isn't it?
It's 500,000. So now Katie has done a lot of academic work on this, says that there are 1,000 instances of etc. So he suddenly goes into a digression. At the end of that, he'll put etc., etc. So it could have been a lot more than 500,000 words. But at the end of his life, he never took anything out. Right. It got bigger and bigger. And very few people have read it from beginning to end. It's not that kind of book. Right. Although Philip Pullman did.
So 1621 was the first edition, right?
First edition. Yeah. And if you get a chance, the first edition, Burton's own copy, is actually in Christchurch Library.
Right. Which is open to visitors, or was.
Yeah.
So it's worth being to.
Thank
you so much,
John. Thank you very much.
Yeah. So we're going to travel in time a little bit because we've gone back 400 years. And now it's a pleasure to introduce Beata Godlewska, who is a consultant psychiatrist and a clinical researcher here in Oxford. And her interest has been in the sort of psychopharmacology and the psychological mechanisms that might be important in depression. So let me see if I can find your talk, Beata. Thank you. And you might just want to stay a little bit back so that people can see your face on there as well.
Okay, brilliant. It's I can only repeat John's words that it's really wonderful to speak to people and also to be able to reach at the same time the audience of more than 250 people, which is a great way of doing it. It was wonderful to immerse in history and to feel, to get the feeling of those wonderful walls that surround us and great ideas. And actually, I've heard that Anatomy of Melancholy is a wonderful reading as well. It's beautifully written.
Speak up a little.
Speak up, okay. Unfortunately, I have to.
We're not showing the whole screen for some reason.
Okay.
I'm seeing it on my laptop.
Right.
The area square.
How do we?
Let's have another go then. There we go.
Is it better now?
That's perfect.
Okay, great.
Sorry to yeah.
That's okay. So unfortunately, I have to take you out from that times and get you back to our current area because I was tasked with presenting how we understand depression now. And I would like to do it in the context of looking back and looking forward, which links to what Freud has taught, that we are what we are because we had certain experiences. A question that I ask is, have we finally arrived? And it is a big question because we've gone a long way to the place we are now. So just a little look at the history of melancholy and depression. It was mentioned already 2000 years before Common Era. So that these are the first mentions of melancholia that we have access to. And then we all know names as Hippocrates or Cicero. Hippocrates thought that melancholia is caused by too much black bile in the spleen. Cicero thought that it has psychological causes. So even then, in this long time ago, people actually were debating whether it's a physical disorder or it's a psychological disorder. Middle Ages went back to the idea that melancholia is caused by demons. So ideas were floating around. And here we have Robert Burton again, that John mentioned in Anatomy and Melancholy. Now, going into the 20th century, in 1921, Emil Kraepelin was the first one who actually took out manic depressions, so mood disorders, as different to what we now call schizophrenia. And they said that those are disorders of endogenous physiological nature. And then the 20th century is a bit of a sort of little battle between psychological theories promoted by Freud, whom we know, Aaron Beck with his CBT, Martin Seligman. And then from the 70s, the prominence of medical models is quite clear, where depression and other mental health disorders are seen as physiological conditions, and as such, they can be treated with medications. The first time term major depressive disorder was used, it was in the 70s, and then in DSM-3, it showed up in 1980. Currently, so going quickly, jumping into the current time, we see depression as a disorder condition that arises from a combination of causes. So it's all biological, psychological, and social factors that actually play a huge role. I know that the audience is quite varied, so I thought I will just say a few words about what depression is. In Europe, we use ICD-10 criteria, but they are very similar to what Americans use in DSM. So the main features of depression are depressed mood, loss of enjoyment, also called anhedonia, and reduced energy and decreased activity. Then there are other symptoms that can, but not always have to be present, like reduced concentration, attention, impaired self-confidence, ideas of guilt, problems with sleep, with appetite. So what comes out of it, it's a really complicated disorder. And the problem that we can possibly all link to, because we all felt sad, we all had bad days, but this is multiplied by thousands of times. And sometimes to the level that people can't move, can't get out of bed, can't eat. So it can be a very, very severe disorder with suicidal thoughts, with suicidal attempts. Now, as a clinician and neuroscientist, I thought I will schedule this talk the way that will touch both sides. So one thing is we really want to understand what this disorder is. And this is where neuroscience takes us. But really what we want to understand to be able to help people. So we need to think about treatments. And I will start by going back and going to current time by showing you that actually treatments have been developed over the past, I would say almost 50, 70 years. But different approaches to treatment of depression were present much sooner. For instance, this is quite interesting that in Belgium and Hiel, people in the community would almost adopt people with mental health disorders to help them on the farms. So it was like the first time when this community psychiatry existed. Now, the question was, have we arrived yet? So I think this study is quite interesting as it tells us very clearly that we have done a lot, but we haven't arrived yet. So it's a nice study, naturalistic study, where doctors were giving medications as more or less as they do in the clinic, going through multiple changes in a scheduled way every 3 months with new combinations of medication, people who would not respond to drugs. And at the end of a 12-month period, 30% of people, more or less 30%, did not respond to any medication. So clearly there is space for improvement, despite us having a lot of different pharmacological and psychological approaches at hand. Okay, so depression, as I said, it's a very multifactorial disease, and it's very hard to actually have one concept. We know that a lot of elements participate, and obviously they are connected together. But somehow it is easier to talk about them separately to show you where we are at this moment of time. They are connected for sure. So going slightly back, not to the 17th century, but only to 1960s, this is when medications, actually current modern medications for depression, were invented. And as it happens often in science, or in psychiatry at least, imipramine was discovered by chance. It was actually designed as an antihistaminic and then used as an antipsychotic. It failed, but then clinicians noticed that people responded very well, people who had depression. So this is how it started, really the history of medications. And then people researched those early medications. And this is how this first theory of depression got born. So the monoamine theory basically claims that depression is a result of deficiency of certain neurotransmitters like serotonin, dopamine, noradrenaline. And this continues, actually, this research is not an old research, it's not outdated, it still continues. It's a very nice, simple, a bit too simple way of explaining how many drugs work. We got a lot of knowledge on the way. We got to know about different receptors, same showing just a serotonergic neuron, serotonergic receptors, but we know a lot about different neurotransmitter receptors and how they work. And this allowed actually moving from those drugs that targeted just serotonin and noradrenaline, especially like this one, which is serotonin noradrenaline reuptake inhibitor, to increase the level of those hormones in the synaptic cleft, to complex drugs, right, like vortioxetine, that actually it's not just a reuptake inhibitor, it acts on many different receptors. So our science, based on those first discoveries, is actually got quite complex. But as I said, 30% of people do not respond to current drugs. So we had to look towards the future. And in probably 90s, people got interested in glutamatergic system in depression. So apparently the glutamatergic system, the glutamate, was recognized as a neurotransmitter only in the 80s, which is surprising in a way, because this is the most abundant neurotransmitter in the brain. And is the major excitatory substance that we have. Basically, it goes everywhere. So the sort of the standard proofs were provided for its participation, like post-mortem studies, genetic studies linking certain genes within this system. Modern science provides us with MRS studies, so it is a way of looking into the brain and assessing biochemicals in vivo. So it's not post-mortem, we can actually put somebody in the scanner, and it's wonderful to see what happens. And those studies also confirmed that the glutamatergic system in depression is hypofunctional. Now, clinical evidence is even more striking. So this is where the future possibly is. Now, the very high drug is ketamine. It's a drug that offers very quick improvement in symptoms that can be observed even in the course of the hours. And well, then the question is how to make it last, because ketamine is a tricky drug. It has to be given intravenously. There is now a nasal spray form, so possibly there can be a continuation of this treatment. But still, ketamine, it is a drug that was an anesthetic from 1964. Then it also became a drug of abuse. So there is always this potential, unfortunately. So we have to know how to manage it, but this is very promising. And the studies really show that ketamine and esketamine, they are useful drugs that are better than oral antidepressants in especially very quick improvement. This improvement can be also sustained for longer with esketamine. Now, how it works. So a bit of cellular science. So basically, ketamine blocks NMDA receptor on inhibitory GABAergic neurons. That leads to disinhibition of the pyramidal cells. We've got a lot of glutamate. We've got another glutamatergic receptor activation, and that stimulates production of BDNF. And BDNF is a neurotropic factor that stimulates synthesis of synapses, increases number of function of synapses. And that's really what is needed in depression. And this action is very fast, so that's why we can see this improvement. This is very different to normal to classical antidepressants that take actually weeks to act. That takes me very quickly to the neurotrophic hypothesis of depression that has been worked on for, again, a few decades. It's very interesting that actually still 20, 30 years ago, we were taught that actually there is no neurogenesis in the brain. So adult brain, what we have, that's it. If our neurons die, that's it, they are gone. However, it's been discovered that actually in the hippocampus, there is new neurogenesis. And ketamine, through stimulating BDNF, is supposed actually to stimulate this process. Chronic use of standard antidepressants also has the same effect. I'm not going to go into the details of it, even if this is a really fascinating area, this idea that our brain can still grow. And going back again to monoamines, so it's all together. So we step back, we step forward. Ketamine also has an action on dopaminergic neurons and leads to an increase of dopaminergic transmission. Again, a lot of work has been done into understanding so which new substances can be used as antidepressants. Obviously, people got interested in this new system because it is a potential. It gives a potential to develop new drugs. And some of those drugs were tested in clinical trials, in particular ketamine and esketamine, but also others. Some are promising, some unfortunately failed. Now, let me take you back again. So we are, again, going back to 1980s. HPA axis. Probably people heard about it. It was quite a known fact that the HPA axis in depression is over functioning. So we've got too much cortisol, there is no feedback inhibition, and this is caused by prolonged stress, for example. And this is a very nice way of producing depression in people. what I wanted to show, we don't have any drugs that would be actually acting very nicely on this system, but go here. But stress actually leads to inflammation, and this is a new area where we can try to invent new medications. There is something that is called sterile inflammation, which is a very nice concept. So stress is perceived by the body as the danger signal. There is no pathogen, there is no activation of the immune system against a virus or bacteria. It's just stress. And it is linked to some extent to this HPA axis overactivity. And then through a number of processes, pro-inflammatory cytokines get released. And that causes a plethora of different effects. So I will just get your attention to the most important things. So again, for a long time, we thought that brain is protected against what happens in the periphery. But now we know that actually cytokines, so inflammation, can get into the brain from the periphery through some leaky spaces in the blood-brain barrier, through diffusion, if there is enough cytokines, but also interestingly through the nerve. So there are chemoreceptors in the vagus nerve that get activated, but even small levels of cytokines. And then this information gets taken back to the brain. And then we get brain inflammation, neuroinflammation. What happens? There is activation of those little guys, they're called microglia, they're basically macrophages in the brain. Their normal function is surveillance, so looking for potentially dangerous agents. They get activated and they produce cytokines, but also substances that possibly we don't want to have in excess, such as free radicals, reactive oxidative species, and so on. Now, I will take you to a different slide to explain this part. This is how we get into neurotransmitters. So again, what we know from 60, 70 years ago. So we can get an excess of glutamate that is toxic to the cells. So glutamate is very needed, but as many things in our bodies, if there's too much of it, that's it. It kills the cells. So basically cytokines activate this alternative pathway for metabolism of tryptophan that doesn't produce serotonin. So we've got depletion of serotonin, which was claimed from the very beginning as one of the main reasons of depression. And then on the top of it, we got production of this quinolinic acid that is toxic to the cells. It also stimulates release of glutamate. Excess glutamate destroys the cells. And the same cytokines can disrupt the dopaminergic function, and especially in the basal ganglia, it can lead to anhedonia or fatigue, which are very interesting features of depression and not well known. We know there is neuroinflammation in the brain. We can do it using modern methodology, PET. There is a radioligand for so-called TSPO, translocating protein density, that doesn't get produced excessively in the quiescent state of microglia, but it gets expressed in abundance when inflammation is happening. And we can detect this substance. And it was seen that the neuroinflammation actually occurs in a lot of regions that can be very important for depression, like prefrontal cortex, ACC, insula, and so on. Now, again, looking into the future, what we want treatments. And this area is actually now one of the very promising areas. So even slightly older, like 7, 8 years old research showed that people may respond differently to medications if they are inflamed or not. And that could, to some extent, maybe explain why 30% of people do not respond to classical drugs. So if there is ongoing inflammation, and then possibly some other sort of treatment is needed. And it was shown, for example, that people who had high level of CRP, they could be helped by an agent called infliximab that acts on the immune system. And also that there was a different response in people to different type of classic antidepressants as a function of this baseline CRP level. CRP is one of those general proteins that tell us that there is inflammation going on. And there's, as I said, there's a lot of research into different aspects of inflammation, which drugs we can use. They seem to be generally working. So those are studies mostly in people who are just depressed, but then there's a lot of studies now going on in people who actually have high inflammation or maybe have a certain gene related to potential action of the drug. So that's the future, definitely. And now I couldn't give this talk without talking about the macro scale. And Macroscale is quite a new research, so it is definitely the present and the future. So whatever I talked about, it happens in the brain and happens in the cells of the brain, in neurons. But the brain then, as we see it, it also has structures and structures that perform certain functions that are relevant for depression. How we do it? We assess the brain now quite easily by scanning people using fMRI, so functional MRI studies where people are given a task and then we see which parts of the brain respond. We can assess the structure, or we can assess the function of the brain also when the brain doesn't do anything at all in theory, because the brain always does something, even in sleep. And this is, for example, one of the functional studies that we've done in the department, and it shows that amygdala, one of the structures in the brain, it lightens up to threatening stimuli, so threatening phases. This is resting state, so different networks of structures that get activated when people just lie in the scanner. What is important is to understand that actually our brains function as networks of structures. So we sort of moved over 30 years from examining the function of singular brain structures like amygdala. And this was a very important period because then we were able to move on to the networks and how it all works. So there is this is a very simple picture, but actually it very much explains how we see depression now. So we see it as an imbalance in control. So we've got those structures that are quite conservative in the brain, like the amygdala. We share them with the animals. They're important. They actually protect us against danger. We will very quickly react to something. We won't be eaten by some predator because we will quickly get away without thinking. Now, this is a very useful part of the brain, but it has to be controlled. So nowadays in the environment, there's a lot of things that can be perceived as danger, but they shouldn't be really, we shouldn't react to them. And this is the role of our frontal cortex to actually correct this bias. In healthy people, there is equilibrium. Unfortunately, people with depression, the structures responding to threat, they are more active. Again, this is a very simple way of seeing it, and the whole network is obviously much more complex, but I think it is quite a good skeleton. Recently, quite a lot of focus went into assessing large-scale network dysfunction, so communication between distinct parts of the brain. And we know that certain networks or structures, like frontoparietal network, it deals with those cognitive aspects, controlling aspects, decision-making. Default mode network is the one that allows abstracting information when there is no task in hand, when there is no danger, and actually making plans, extracting information from the past to make use of it in the future. And then there's affective network responsible more for emotions and other attention networks. And all those networks, they work together to create a healthy brain that can adapt and respond to changing environment. In depression, the communication between those networks is, unfortunately, is disturbed. As I said, a lot of research goes into it now. So this is one of the definitely future tasks to understand how we can use neuroimaging. And one thing is, so we know that using certain tasks, we get certain activation of the brain. We don't have a biomarker of depression. That could be one potential field for it. There is no structure currently that actually can be a biomarker yet. So we know that there is, it functions in a certain way. The person definitely has depression. But there's a lot of work going into neuroimaging and treatments. And that's quite interesting. There's one structure that's called anterior ACC. And it's been shown that if it's overactive, then people have a better chance to respond to our classical antidepressants. Again, a lot of work goes into in order to get a lot of specificity about this. So we would like to know really that there is some pattern of activation in the brain that will predict response to a certain medication. For now, all our clinical attempts to treat people are based on trial and error. So we know which medications are most successful in most people, but really it is a stepwise process. So it would be fantastic to say if people say, if I go back to this first slide, 12 months of waiting to actually do the brain scan and then know that this person should get this drug, not this one, and they will respond to it. Now, neuroimaging can also help in drug discovery. So it's potentially a very important tool for this. So if we know that the brain responds in a certain way to the drugs we know, we can also test new compounds based on it to then say whether this drug should be tested, should go into the phase of clinical trials. So it will save potentially a lot of time and energy. So that's another area of research here. Definitely the future, there is the role for machine learning. And now we are able to use computers, quite powerful computers, to extract certain patterns of activation in the brain that will tell us something that we want to know. So we can ask questions about depression. Is there a pattern of the brain that can tell us that somebody has depression? Is there a pattern of brain activity that will tell us that somebody will respond to a given treatment? And like this study, this approach allowed to identify 4 types of depression that actually have different sets of symptoms, different combinations of the same symptoms, and also predicted response to TMS. So there is potential practical application. Unfortunately, this is just the start of it. But that's why this is the future. The future definitely is also sits in novel neurostimulation treatments. And we do a lot of research on TMS, transcranial magnetic stimulation, TDCS. So both approaches, they use current to activate or inhibit parts of the brain. There are studies on DBS, which is internal stimulation, which is a very it's a very difficult, it's a surgical procedure where of implantation and activation of again certain parts of the brain, and those approaches are very actually are good. So studies have shown, meta-analysis, that response rates are much higher than in like sham treatments where people don't get any stimulation. I would like to just mention, going very much back to the past, ECT, electroconvulsive therapy, that sits well with those novel treatments, is still a very good approach, and we use it. We use it in a very humane way, so people get anesthetized, and people respond quite well to it. It got bad press, so if somebody hasn't seen the famous movie with Jack Nicholson, One Flag Over the Cuckoo Nest, I would recommend it. This is a good approach. Again, I'm finishing now because I think my time is very much up. But just to say, psychedelics as treatments for depression. The smiling guy here is Timothy Leary, who's also a person from general culture now, who promoted psilocybin. And psilocybin actually acts on the brain. No surprise here, but there's been studies showing that it may be a potential antidepressant. The future is combining psychology and biology. So I didn't speak a lot about psychology because that's a neuroscience symposium, but psychological treatments work on the brain. What we do, especially CBT, was examined in this context. It actually does produce changes in the brain that act the same as medical treatments. And we don't know why some people would respond to this treatment, some to this. So we try usually both approaches. And here we use also psychology and biology to explain why there is this delay in antidepressant action. So combination of biological effects, but also what has to happen is a positive shift in emotional processing in the amygdala, so it doesn't see everything as a threat. Psychological therapies, as I said, are still used. And before I say thank you, let's not forget about genetics. That also lies at the background of all of it. Okay, thank you very much.
Thank you, Beata. Yeah, and we've got some questions, I think, in the chat. Let's see if I can find some of these. Could there possibly be scope for machine learning to be introduced to young people, adolescents, who have predisposition to depression or mental health disorder as a form of early intervention? Is that really blue sky stuff?
I think it's still far away from us. It's just the beginning of machine learning technologies, but it is a very good suggestion. It would be really nice to be able to identify people who are predisposed and who can develop depression, at least to monitor them and maybe to apply some non-invasive approaches like CBT to actually to help them to see the world differently.
Yes, I guess the question is about finding people at risk, at predisposition.
Yes, exactly.
Here's another one. How do you analyze if certain treatments are working? Do you monitor the neurotransmitter or the condition of functional structures of the brain, or just how the patient is?
Yeah, so I think this is one of the problems of psychiatry. There's this big divide still between a big gap between what we know about the brain and how it functions and all the theories they showed you, and how we actually treat people in the clinic. So we still do person-to-person assessment. We still give medications.
I would hope so, yeah.
Yeah, we do. Yeah, even in the pandemic era, we're able to talk to people. We give medications based on our clinical knowledge of what works best and certain suggestions, certain guidelines. We do not have any indices, biological indices of working, medication working. We don't do it as a standard thing for sure.
Okay.
It's a shame.
If there's anyone in the audience here who would, go ahead. Shout out there.
I had a question about ketamine. From my understanding, people have looked at the metabolites of ketamine and showed it has similar antidepressant effects acutely, but they don't have the same efficacy at the NMDA receptor. So I'm kind of wondering about where are we at in our confidence in that glutamate hypothesis for ketamine?
We've got some confidence in ketamine, definitely based on the clinical results of what we see in patients.
So of those 30% of people who are treatment resistant and you give them ketamine, how many of that group will respond?
I think this is probably a hard question, but quite a few with treatment-resistant depression. I would say probably yes. Yeah, so that's quite a good thing. The problem with ketamine is that it's difficult to administer. It has to be done in the setting it's IV, and also this addictive potential. So,
but your question is it really ketamine or its metabolites?
My question is it really glutamate and NMDA receptor, given that the metabolites can have efficacy on their own and they don't?
That's what we think at this point, and obviously it can change. We know that glutamate definitely changes, dopamine changes, but also possibly ketamine can act through opioid receptors, and there was one study actually showing that its action was abolished when opioid receptors were blocked. So we don't really.
So it's a good point. It may not be through the.
So it may not be true, but
okay.
People tend to think it is for now.
But clearly we've come a long way since Burton in terms of mechanistic thinking. So thank you for taking us through that, both in terms of neurotransmitters, inflammatory markers, and also the systems level.
Thank you.
And psychology is part of neuroscience, being included.
Oh, absolutely, absolutely.
Okay, thanks a lot, Beata. Yeah. So our next talk today is from Zoltan Molnar, and let me just find Zoltan. And Zoltan is Professor of Developmental Neuroscience here at Oxford. Sorry, let's find your one. I'll let you find it, Zoltan. Can you see it on there?
Thank you.
Great, yeah. And he's a great enthusiast. The students here have see lots of Zoltan's enthusiasm, but he's also really interested in the history of neuroscience. And today he's going to focus on Thomas Willis and his contributions to clinical neuroscience. Thank you, Zoltan.
Yeah, so on the screen here.
Why that's happening? Is that okay? There's a delay, I think.
That's not going to on Zoom.
No, I think it's a delay. Here we go.
But
Ah,
We've never seen that in practice.
yeah. Let's try it. It worked with a. Let's put that off. Let's have another look.
Are we going to present projector mode?
Sorry?
Go to Zoltan's talk at the bottom. Get projector mode on the first slide. Now go and try and share it.
Not sure that does much. No. Okay, I'll do one more go and then if
Maybe we should also, yes, so if get it into it's not objection mode.
do you want to come and do it, Nick?
So if we get into projector mode there,
I think that just does what. Yeah.
now we should now without any, of course, yeah, such thing.
No, you can't do that.
Maybe we should close some of these talks down, might make it easier.
So sorry, Zoltan.
No, I'm
The,
sorry. I wish I knew how this is operating.
I'm going to close these other ones down and let's say we've never had that before. Yeah. Sorry,
But then you can't share that.
you can't share that way. Yeah, we tried that.
Okay.
Okay.
Yeah.
Brilliant.
That's perfect.
Thank you.
Beautiful. Thanks, Zoltan.
Thank you very much, Masood, and also Nick, for this opportunity to talk about Willis. As you know, Willis was born 27th of January 1621, exactly 400 years ago. So this year we had a lot of events, and I want to draw your attention to the St. John's online exhibition, which we put together. We also have a huge website dedicated to Willis at the DPEG website. And also there will be one more event I want to draw your attention at the Royal Society on the 18th of November. In fact, this is the day when he was elected Fellow of the Royal Society. So that's why Alastair Compson, who just published this book, decided to have this meeting on that day at the Royal Society. So its registration is free. So in the next 10 minutes or so, I can only give you a flavor of Willis, but I think it is important that the new generation will hear about Willis. And by the way, Alastair owns about 80 books published by Willis. So he has the largest collection in the world of Willis. So I would like you to come back with me to the 16th, 17th century and just try to think about what did we know about the world at the time. So we started to understand where the continents are. There was the first textbook of chemistry published, De la Pyrotechnica. And it was the time when Copernicus published the heliocentric view of the world. Our view of the brain was very rudimentary. It was based on Aristotle's ideas of bodily humors and Galen's observations of fluid-filled ventricles. And this idea of cell doctrine was dominating the medieval literature, and everybody kept copying these pictures. Probably they have never seen the lateral ventricle, including Leonardo da Vinci. At early stages of his career, he was drawing these ventricles, but then he changed his view by injecting molten wax into the ventricles of an ox brain. And then he changed his view, and you can see that you see the two lateral ventricles, you see the third ventricle, the aqueduct, and the fourth ventricle, and it's very similar to a human cusp we use to teach second-year medical students for neuroanatomy. So things were changing in anatomy, and it's because of the experience. Anatomists no longer believed what they read. And usually anatomy teaching there was a teacher who was reading Galen's work. Then you had a sector who was dissecting, a barber. And then there was one person who was actually pointing, the ostensor, at the different parts of the body. And as you can see, medical students did not pay any attention to any of this. and what changed was, at the time of Vesalius, is that the professor started dissecting and wanted to see everything with his own eyes. And that's why there was a huge revolution in anatomy. All these lecture theaters were established around the world. And also Vesalius published the Humani Corporis, which is an amazing book of anatomy. In England, Henry VIII passed the Act of Parliament that enabled the College of Physicians to regulate the practice of medicine. So you had to have a license to practice medicine. And also, Elizabeth I gave the college of the right to collect the bodies of 4 hanged criminals each year for dissection. And Oxford also had this right. The Tomlin's Reader of Anatomy could dissect 14 executed prisoners a year. This is the time when the colleges were established. Oxford already admitted 400 students to study. So it was quite an academic powerhouse in those times. And this is the time when Willis was born. He was born in Great Bedwin, which is not far from Oxford. He was born at Farm Lane in this actually, I drove there just to have a look, and this didn't change too much. It's the same size. Of course, you don't have these solar powers in 1660s, but this is the house where he was born, Ivy Cottage, or the castle, as they call it. But he only spent a few years there before he moved to Oxford because Rachel, mother, inherited some land and this house, Fairy Cottage, in North Hingsea. And this is where Willie started to go to school, walked every day to High Street, and he received classical education. And then he was admitted to Christ Church, where he matriculated to continue with his studies. His status was a servitor, so he had to do some work to pay for his fees. And his task was to help the wife of the dean, Mrs. Ives, and this is how he learned a bit of iatrochemistry and medicine. And this has changed his interest to medicine. And perhaps this was the most important education he received from Oxford. I always tell this to students that Willis became the founder of clinical neurology and neuroanatomy in spite of the Oxford education. He was spared from the education. He didn't have to study medicine for 14 years, and he could start practicing medicine because the Civil War broke out. He was at the side of the royalist troops, so this was not the time to study, really. Cannonballs were fired into colleges. My own college, St. John's, received this cannonball from the rebel forces of Cromwell, and the college kept it in the library. And basically, Willis couldn't study. And he was very lucky because in those days, you could get your degree directly from the king. And since he was royalist, Charles I gave him the medical doctorate degree just before he was beheaded. And Willis could start his medical practice, even during the Cromwell years. And he experienced quite a bit before the Restoration. So this is his medical degree. And then when Charles II came back during Restoration, and then again, Willis found favor and he was elected Sedleian Professor of Natural Philosophy. By that time, he had a huge clinical experience. And he started publishing he lived in Oxford all his life with the exception of the last 9 years when he moved to London to get more money. And he planned several other books, but then he died in 1691. So we know quite a bit about his clinical practice from his notes. He didn't want to publish these case books, but they found it. And from this, we know where he practiced, quite a large area. They had to ride out to these market towns like Abingdon, and they had to compete with doctors, other young doctors, and also with charlatans who didn't have a degree. And we know about his patients from this case book, and look at the variety of the cases he was describing. And we also know how much he charged for this. Some of the descriptions of these conditions, just amazing. You could still use these for teaching today. So this was his strength. He didn't believe anything he did not see. He had to see himself. And he didn't work on his own. He had a fantastic team surrounding him, including Christopher Wren, the architect. His student, Richard Lauer, was extremely prolific. Then we had the chemist, Robert Boyle. William Petty was his friend, who was the Tomlins reader of anatomy at Christ Church. So they dissected as a team together. And although the books were published by Willis, he always acknowledged this help. So he was working and dissecting with William Petty. And initially, they both struggled financially. But then after the resurrection of an executed prisoner, Anne Green, who was probably not hanged properly, they produced this leaflet explaining how good their medical practice is, because they can actually resurrect an executed prisoner. Their career took off, and initially they were dissecting in Petty's residence, which was off from High Street in this building. They could move to a more fancy house. So this Petty's residence is still here in Oxford. It's difficult to find because it's in a narrow alleyway, and now it's hosting the Chiang Mai Kitchen, an excellent Thai restaurant, which I recommend to all of you. So after this Anne Green episode, Willis could afford to move to Merton Street. So his residence was opposite the entrance to Merton College. And there is a debate whether he actually lived in number 3 or number 4. According to Alistair Compton, he lived in actually number 3, but the plaque was put in number 4, where he actually worked. So the two buildings were connected to each other, and it's called the Beeham Hall or Beam Hall. Beeham was vice chancellor at Oxford who owned a building on that site before Willis. So it was a teamwork, and they described their observations extremely well. They used fixative. Robert Boyle advised Willis to use port and vinegar to pickle the brain so they could look at them longer. And also they interacted with all these other researchers at Oxford. Some of them are from Wadham. And basically, this was the foundation of the Royal Society. So this group of people founded the Royal Society here in Oxford. Now, Willis's legacy is considerable. And when we teach neuroanatomy, we have to use all these terms he gave to us, and we still use them. His descriptions are superb. And also the illustrations from Wren are excellent. And he became extremely famous because of the Willis circle, and every medical student have to learn the Willis circle, which is an arterial circle at the bottom of the brain formed by the two carotid and also the two vertebral arteries. And then you have the three pairs of the cerebral arteries from here. But actually, I will show you that he wasn't the first to describe that. But his illustrations were so excellent that everybody started calling it the Willis circle. He described all the cranial nerves, but his numbering was slightly different. So if you look at the illustrations, you can see that all the nerves were there. And look at this amazing drawing of the autonomic nervous system, where you can see the paravertebral sympathetic chain, you see here, and you can also see the vagus nerve, you can see the plexus cordis. And in fact, he conducted experiments where he ligated the vagus nerve and looked at how it affected the dog's heart. So way ahead of his time. Some of these diagrams are abstract, and actually they're anatomically correct. We could use them for teaching even today. One of his first books described fevers, excellent description of malaria. His second book, A Cerebral Anatomy, is the first book which is using the term neurologia or neurology. And neurology came from the translation by Portage later on. And this is the book where you have the circle of Willis. But as I mentioned, these illustrations were done by Wren and also his student Lauer. And he always acknowledged that this is a teamwork. And he gave credit for Lauer for the anatomy, and also Wren for the illustrations. So these books were from Willis, but this is actually a result of a considerable teamwork. So as you know, Wren also is the architect behind the Sheldonian Theatre and All Souls College, and also the St. Paul's Cathedral. So he is an incredible architect. So the circle of Willis was described by several people, but Willis emphasized the functional role. So Fallopius described it before him, Casserius, and many others. You can see that the circle is clearly present. The vasculature is a bit more hairy in this drawing, but nevertheless, it's correctly depicting the circle. And also Johann Jakob Wepfer in Switzerland, he even described the functional implications before Willis. But nevertheless, we call it the circle of Willis because of the excellent illustrations. That's why it's great to know a little bit about the history of Willis for every medical student. So he clearly described that nature had provided a sufficient remedy against the risk of apoplexy. And this circle is not present in everybody. There are huge anatomical variations, but basically he got this absolutely right. His classification of the cranial nerves were in use for 150 years after his death. The person who actually then changed the numbering was a medical student from Göttingen, Sommering, and now we use this. It's not perfect. I could change this even now, but this is what we have to teach now for the nerves. He also described epilepsy. He associated the cause of hysteria with the brain, which was very progressive at the time. And he also advised his colleagues through correspondence, and some of these letters are here in Oxford at St. John's College Library. So we know how he advised some of his colleagues, and he showed great interest in the outcome of the treatment. So he was a fantastic mentor, clinician, researcher, educator. He could combine it all. But for me, and this is my last message about Willis, for me, the most important contribution Willis had, that he associated the higher cognitive functions with the brain, with the cerebral cortex. He compared different species, you can see here a human brain and a sheep brain, and he noticed that the brainstem was very similar in proportions and in size, but the cerebral cortex had more convolutions and it was much larger. So this was one of his arguments. The second argument, he looked at his patients and their brains after they died. So he had the case history, and he noticed that this boy, whose brain is depicted here, had signs of epilepsy and learning difficulties. And there were other infants from the same family. So he even thought about the genetic origin of these developmental disorders. And probably this boy had a polymicrogyria syndrome, which he described in 1664. And based on the comparative work and also looking at the patients with brain shape abnormalities, he concluded that the cerebrum is the primary seat of the rational soul in man and the sensitive soul in animals. It is the source of movements and ideas. So he got this absolutely right. The cerebral cortex is the seat of these higher cognitive functions, language, memory, episodic memory, initiation of movement, and all these higher cognitive functions, and he got that absolutely right. So that's why I think it's important that all new generations of students, they learn a bit about Thomas and Willis, and I think we are all very proud to be here in Oxford where he worked 400 years ago.
Absolutely fascinating. So even if you're not interested in history, I think you'd be interested about the history of neuroscience through Willis's eyes. And if you're interested in reading more, Zoltan has written a very nice essay in Brain a few months ago, which is a really sharp insight into all that you've heard here.
Masood, thank you as the editor for commissioning that paper.
No, not at all. Let me just see if we've got any questions here. Okay, no, amazing, thank you.
Thank you. Thank you very much.
Okay, thank you very much, Sultan. So I was wondering where to take this, in terms of time travel 400 years back, and Willis's contributions are so manifold, but I thought what we could do is to link his contributions to neurovascular anatomy and cognition. As Zoltan said, he was probably the first to think about the cerebral cortex in this way as a seat of cognition. So I thought, who better than to ask Michelle Veldsman? Are you going from your computer? No.
No, from there.
Yeah. Michelle Veldsman, who is a research scientist here, and Michelle's background is in psychology, cognitive neuroscience, and neuroimaging, and she's had a particular interest really in the effects of stroke and also vascular disease on the brain on cognition. So let's see if we can get. By the way, thank you, Kengo and Verena, for helping me out on that one. Yeah. Also to be able to see it.
Okay.
Can you see it?
Yeah.
Oh, okay, maybe you should do. Oh, great. Brilliant.
Oh, brilliant. Okay.
Thanks, Michelle.
Thank you. And thank you to Mr. and Nick for inviting me to speak and organising all of this. So my talk really is mostly looking forward and putting together everything that we have now to understand how vascular cognitive impairment works, what's the basis of it in the brain, and how it interacts with other dementias. Dementia and vascular impairment has a really long history, and as most of you know, today we really lack disease-modifying treatments for most dementias. And for me, the future really is about the increasingly high-resolution brain and body imaging that we're able to do now. The kind of computational methods that we have, including artificial intelligence, has been spoken about earlier, and also the combination of genetics, lifestyle, medical information, all of that data that we have now in entire populations. Sorry about this is the future. Okay, so I'm not going to touch anything there. I accidentally had a finger touch on there. Okay, so vascular cognitive impairment basically refers to cognitive impairment that's caused by or associated with vascular risk factors. And by that, we of course just mean anything that impacts the blood supply to the brain. And it really exists on a spectrum. So you have from mild vascular cognitive impairment all the way through to major, where you have really problems in day-to-day life with cognition, as is the case in vascular dementia. But because there are just so many ways in which the brain vasculature can be affected, there are multiple ways that you can end up with vascular cognitive impairment. So it might be due to occlusions or blockages in the arteries in the brain. It could be bleeds or hemorrhages, often after stroke, ischemic or hemorrhagic. There's dementia, post-stroke dementia, and then there's hereditary conditions as well, like CADASIL. And then that's not really forgetting mixed dementia, which is probably a lot more common than we think. So with this in mind, I just hope to convince you of three things today. One, that vascular risk factors are associated with subclinical cognitive decline and aging that we can detect at a population level. That the obvious MRI markers are the tip of the iceberg in terms of vascular degeneration, and that vascular pathology contributes to really a cascading mix of pathologies that are occurring on a background of aging. And that's really important for our thinking about how dementia arises and progresses. And why does it matter? Well, as you know, we lack these disease-modifying treatments for many dementias. And really the focus now is on finding the earliest possible markers in the brain that might underlie later cognitive impairment. And some of those undoubtedly will be vascular in nature. Vascular risk factors are really important to understand because we need to know when to intervene, and it's often decades before we see anything in the clinic. And then finally, you know, in the search for disease-modifying therapies, it's important to account for this vascular component, because those disease-modifying therapies might only, by their nature, modify the pathology of that disease, for example, Alzheimer's disease, and that might not treat the other vascular components that have an impact on cognition. And the important thing about vascular risk factors is that they're modifiable, they're treatable, they're mostly preventable, and treating them can actually delay or prevent dementia, and any delay or prevention to dementia has a massive impact. So over 2 years saves 12.9 billion a year, over 5 years 21.2 billion a year, and you know, some of that money would hopefully be then directed towards finding disease-modifying therapies and treatments. And also a 5-year delay can also reduce the number of people living with dementia by almost 500,000, and the number of people who are caring for people living with dementia. So to speak to this first point, that there are vascular risk factors associated with subclinical cognitive decline and aging, I'm talking here sort of the really subclinical cognitive impairments that are very difficult to detect and require quite sensitive measures. And this is usually seen in sort of population-based communities. And you've probably heard the phrase healthy heart, healthy brain, and that is because many of the vascular risk factors, cardiovascular risk factors, are the same as the cerebrovascular risk factors. So anything that's affecting blood flow generally is bad for the brain. And that includes things like physical activity, obesity, lack of physical activity, sorry, obesity, hypertension. And it's notable that the majority of the modifiable risk factors for dementia are sort of vascular in nature, and control of those could prevent or delay 40% of dementias. So if we take hypertension, for example, we know that one of the strongest predictors of small vessel disease is hypertension, and that's one of the pathways to vascular cognitive impairment. And we've known for a long time that the control of hypertension can reduce the risk of dementia, and the use of antihypertensives also reduces the risk of dementia. And my own work, what we've seen is that even small elevations in systolic blood pressure can have an effect on cognition. So for the first time in history, really, we have access to really detailed demographic, health, genetic, and lifestyle data in hundreds of thousands of people. And we also have the computational power to be able to analyze in detail brain images alongside all of that data. So here I'm showing you data from 22,000 people from the UK Biobank, and it shows this really continuous relationship with systolic blood pressure, executive function with systolic blood pressure, sorry. So people not on antihypertensives have this continuous relationship where as you have increases in systolic blood pressure, you're worsening executive function. But when people are on antihypertensives, either diagnosed as having high blood pressure, there's this stable range in which actually their executive function is quite stable until it then around 140 starts to decline quite rapidly. And also you can see in general their executive function is lower than those not on antihypertensives. And that seems to be quite specific to midlife. So it's important to treat people in midlife to get control of hypertension at that stage. So then the question becomes, what might underlie the cognitive impairments that we see associated with with hypertension, sorry. MRI is the standard in the diagnosis of vascular cognitive impairment, and many vascular pathologies are actually visible on MRI. And by far the most commonly investigated MRI marker is white matter hyperintensity. So there have been, over the last 30 years, thousands of papers that have investigated the relationship between either the presence or the burden of white matter hyperintensities and the risk of dementia or cognitive impairment. So white matter hyperintensities, I should say, are these regions which are either around the ventricles or in the deep white matter that reflect usually vascular damage. And it's quite complicated with white matter hyperintensities because of the fact that age is the strongest predictor. So at age 64, around 21% of people have evidence of white matter hyperintensities, but by 82, around 94% of people have them. And you can have quite a high load of white matter hyperintensities without any impact on cognition. So that complicates the story further. But we know it's not just a benign feature of aging because their presence triples the risk of stroke and doubles the risk of dementia. And it's also associated with a faster decline in global cognition, executive function, and speed of processing. And for a long time, we thought that all white matter hyperintensities were the same and just reflected vascular damage, and that they always fell in the same sort of stereotypical pattern around the ventricles and in the deep white matter. But now we can use population level data to try and unpick the spatial distribution of white matter hyperintensities a little more. Because we have data in thousands of people, we can now look at the individual risk factors, things like hypertension or obesity, without them influencing each other, because often these things are comorbid. And what we see is a sort of different spatial distribution depending on the different vascular risk factors. So here you can see the probability of white matter hyperintensities in 13,000 people for the different vascular risk factors. And waist to hip ratio, ApoE status, and hypotension have different spatial distributions. And in fact, it's waist to hip ratio that the relationship between cognition and white matter hyperintensity load is actually mediated by waist to hip ratio and not hypertension, which might be what you would think it is, because hypertension is the most, the strongest predictor of the load of white matter hyperintensities. So this just emphasizes the point that the obvious MRI markers might just be the tip of the iceberg for vascular degeneration in the brain. And there probably are better markers that we can get from MRI. So in the UK Biobank cohort of 22,000 people, I showed you earlier, showing this relationship between executive function and systolic blood pressure. We looked at different brain markers, the obvious white matter hyperintensity, but then also some microstructural measures and the volume of regions of a frontoparietal network that we know is important for executive function. And we did this using structural equation modeling, where we can look at a number of different variables and see how they interact with each other. And the important thing here, and quite a complex model, but the important thing to note here is that frontoparietal white matter integrity and frontoparietal gray matter volume have similarly predict executive function. So they are important predictors of executive function. These are standardized beta estimates. So you can see that they're a similar magnitude, and they are an important feature of executive function in aging. And when we look at the relationship between white matter hyperintensity load and executive function, what you find is that there is a modest relationship around 0.2. But in fact, when you control for white matter integrity and gray matter volume, this relationship really disappears. So it suggests that actually a lot of the relationship between cognition and white matter hyperintensity load is actually explained by the integrity of a distributed brain network. And even in a stroke population where you have very obvious, vascular marker, a stroke macroscale marker there that you would think would be very important for executive function. I should say around 40% of stroke patients, regardless of where their stroke is, have long-term executive impairment. You find that actually the white matter integrity of this frontoparietal region is a better predictor of executive function than the stroke severity itself, and that's including the stroke volume. So this again just emphasizes the importance of these microstructural measures beyond the kind of obvious vascular markers like white matter hyperintensities. So finally, I just want to talk about how vascular pathology contributes to this cascading mix of pathologies on a background of aging. So we know that there are several large pathological studies that show that actually mixed dementia is the most common type of dementia in aging, and that vascular pathology increases the likelihood of dementia at any level of AD pathology. And then conversely, small vessel disease has an additive effect on neurodegeneration. So in people with AD pathology, you get this decreased perfusion and blood-brain barrier leakage that likely results in vascular pathology that then adds to kind of mixed picture of dementia. And here I just actually relatively simplified picture of how these things may interact. So here, Alzheimer's pathology and then vascular pathology, all interacting in this really complex cascade that results in quite a mixed picture. And because these disease-modifying therapies do still elude us, I think it's very important to take into account, the additive burden of vascular pathology. And then, of course, you add to that the sort of genetic vascular risk, cognitive reserve brain resilience. And this all sounds like it's very complex and difficult to figure out, but the positive thing is that we now have this population level data, and we have this incredible imaging, and bringing all of this together can really get us closer to understanding this complex problem. And of course, vascular risk is not the only solution, and it's not going to, solve all dementias, but it is a really important part of the picture. And importantly, we can address it now, so we can put in place strategies to manage and control vascular risk factors. So I really, just to conclude, hopefully I've given you a brief picture of the fact that vascular risk factors are associated with subclinical cognitive decline, and that we can see even small elevations in blood pressure can impact cognition. MRI markers are like white matter hyperintensity. These are probably only the tip of the iceberg. There's also a lot of breakdown in these distributed brain networks that are underlying cognition. That's very important, and that we don't necessarily see visibly on MRI. And that really vascular pathology is a really important part of the picture in terms of mixed pathologies, which are actually probably quite common. And then just remind me to say thank you.
So we fast forwarded to the current time in terms of what Willis's contributions might have led to in terms of neurovascular and cognitive function. Any questions here? Brief questions for Michelle? No? Okay. Let's see if there's anything on the chat. Lots of things on the chat. Okay, so someone says, this is really amazing work, practical ways to reduce dementia prevalence. Do you know of any work being done in low middle-income countries to address these factors, risk factors, like obesity or hypertension?
Yes, definitely. There are a number of large-scale studies that are trying to do this. And I think it's really now becoming clear that we actually need to take into account a lot of the social factors as well, because I sort of mentioned briefly there, cognitive reserve, and one of those modifiable risk factors is actually education and sort of remaining engaged. So that's something that needs to be addressed as well, because one of the things that we find is this idea of sort of brain resilience, where you can have a lot of pathology without impacting cognition, and a lot of that is to do with this cognitive reserve. So that's a really important part of the picture, I think.
Yeah, no, that's very important, isn't it? The variation in resilience. One final one, curious about whether there's a point of no return with modifiable risk factors, i.e. if patients have more than 10 years of untreated hypertension by midlife, are we beyond repair? Is there any evidence of that?
That's an interesting one.
I'm not sure about the evidence for that. Actually.
Yeah, I don't know if we have a lot of evidence for that, to be honest. And I mean, partly one of the issues is that a lot of other diseases are cropping up that's associated with those things. So a lot of these risk factors are the same risk factors for cancers and for heart disease and all of those problems. So it's actually quite a difficult issue because of this idea of sort of multimorbidity.
Okay, we'll have to stop it there. Thanks so much, Michelle. Thank you.